Hi, welcome. Welcome to the Linnean Society of London. How many people is this their first time here? Oh, wicked, that's great. So my name is Leanne Melbourne, and I'm the Events and Communications Manager here at the Society. So I'm just going to go through a bit of an intro about the Society and what we do and how this day came about. So we are the world's oldest active biological society, and we were founded in 1788 by Sir James Edward Smith. And we're based on Carl Linnaeus' collection. So we have them stored in the vault downstairs, and we do tours once a month where you can go into the vault and have a look at his collections. So he's the founder of Modern Taxonomy, and Systema Natura was one of his books that he wrote. So we have over 2,500 fellows, and anyone who has an interest in natural history can become a fellow. And we have an international fellowship, so the majority of our fellows are here in the UK, but we have fellows in Australia, US, India, all over. So today, this has come out a bit weird. Yeah, okay, so today's conference kind of stemmed out of, during Black History Month in October, we decided that we wanted to look at the contributions of ethnic minorities in natural history, and I wanted to highlight our fellows, and it was very difficult. So partly because we don't actually keep ethnicity data, so we don't actually know who of our fellows are in the UK who are ethnic minorities, but it there wasn't really a lot out there, and so I kind of broadened out the reach. Instead of looking at just fellows, I was looking at just contributions in general, history, past to present, and yeah, I was struggling a bit. And then I came across John Edmondson, who taught Charles Darwin taxidermy, and I came across this as well. This is from, it's not on, here we go. This is a painting by an Antiguan John Tiley, and he worked with a Scottish botanist, Alexander Anderson, in the St Vincent Botanical Gardens, and so he did quite a lot of paintings, and I'm slightly obsessed with his paintings. I put them everywhere, so it's like on the programme's brochure, you've got postcards. Yeah, I love them. I've even slipped it into the anniversary meeting agenda for the fellows, so yeah, so and then it got me thinking about the hidden contributions, things that are hidden in plain sight. Basically, we see them every day and we have no idea where the actual contributions are coming from. And I was having a chat with Anjali Goswami, and so we kind of felt, last year we did this Women in Science conference looking at contributions of women, like let's do another kind of day looking at diversity and ethnic minorities. So that's where the day came from. So I thought what I would do next is kind of set the scene a little bit, and I've done a little bit of looked at some stats. Like, it's actually really difficult to find statistics on ethnicity, but I found a few things. So first of all, let's look at the headlines. These are like the headlines that I looked when I Googled ethnic minorities, STEM, science. So you've got ethnic minorities underrepresented in science. You've got British universities employing no black academics in top roles. You can see they're all from The Guardian. Only The Guardian seems to report these things. UK universities make slow progress on equality data. So those were kind of the storylines, the headlines that I was seeing. So I decided to try and look at the actual stats. So this is kind of from the Equality in Higher Education report. So what we can see here is, so this is like the census. So in England and Wales, 86% of the population is white, and ethnic minorities make up 14%. And when you look at students, how that's represented, is actually represented quite well. So we've got 26% ethnic minorities students compared to 74%. And then you can see the breakdown in the ethnic minorities here, so mainly Asians, and then blacks, Chinese, and mixed. So when we start to look at STEM, so we've got STEM subjects here. You can see all STEM subjects, 76.3% students are white, and 23.7% are of ethnic minorities. And then if we look at medicine, it's even, you've got 35% of the students studying medicine are of ethnic minorities. And biological sciences is quite good as well, with 20.6%. But then we can see the breakdown here, especially medicine, the majority of the ethnic minorities are Asian, and then you've got, there's a difference between the different minority groups. And the same in biological sciences. So that's all students. So then when we look at undergraduate versus postgraduate, you get, that's when the drop really is quite large. So from 20% of students studying biological sciences undergraduate, And then that drops to 13% for postgraduate. And you can see the same for medicine, so 37% drops down to 26.6%. And then you can see the difference as well between the minorities. You get even larger drops within like the black and mixed groups compared to Asians. So that's undergraduate to postgraduate. It gets a bit difficult to start looking at staff because the cost centres don't really match up, but we can kind of have a bit of an idea. So we've got here STEM sciences, all academics, so these are nearly 90% of academics are white compared to 11% ethnic minorities. And then when we look at professor and non-professor, it's pretty much quite similar. And then when we kind of look at different subject categories, so the different cost centres, you can see that clinical, this is all staff. Clinical medicine, we have about 15% of staff are of ethnic minorities compared to 85%. And then it's even lower for like natural history, so environmental sciences, geography, for marine sciences, where, I don't know if you can see that number, it's 3.3% of staff are of ethnic minorities and 4.6%. So you're seeing this large drop. So you're seeing a drop from undergraduate to postgraduate to staff level. So that's why we're here today, to kind of discuss things that, what's going well so far, what we can do better, and I'm really excited for today. I hope you all are. And so first up, we have Miranda Lowe talking about her experiences and the history and the contributions of ethnic minorities to natural history and how it's influenced her experiences in the museums today. Thank you.
Can everyone, yeah, you can all hear me, because I can hear myself. Well, welcome. So, thanks for that introduction, Leanne. That's great. So my talk today is about diversity in the face of biodiversity. So I'm a scientist at the Natural History Museum, well, scientist and a curator, so I wear two hats. But, you know, I spend a lot of time, and the other scientists along with me in that institution, looking at things to do with the topic of biodiversity. But as Leanne pointed out, there's a big problem with diversity of people like me within that institution, and diversity in terms of the narratives and the stories that are told within those institutions. So this is a biodiversity bit. So these are the kind of things that I actually work on in the institution. So crustacea, there are corals, but I think these pictures are mainly of crustacea. So we live, you know, and we examine and we study the biodiversity in the natural world. So that last picture was a blue swimming crab. But not many people of colour in days of old, many years prior to my being at the Natural History Museum, were able to tell their own story, tell their narrative in their own way. But what I'm very lucky to have been able to tell my story in the way that I want to, and to have that embedded in the history of the Natural History Museum. So I took part in a project that was run jointly with our Arts and Humanities Centre that existed at the time. I think it was probably about 8 years ago. And it was a joint project between the museum and Kingston University. It was called Museum Lives. And as you can see in the pictures along the top, so there were sort of more than 4 hours of me talking about myself, and I don't do that very often. I would say I'm doing it again now because I'm being filmed for a documentary about me as well, so I'm doing it again after such time. But what was really interesting about this project, that I was able to talk about my personal life before I joined the museum growing up, my time in the museum, and what I saw for my career in the future. And it was all on my own terms, so whatever I said has been documented, and will be there forever. So it's in various media. and it's stored in the archives. And what was interesting, I didn't, because usually when I'm doing my research, I'm looking for people in the archives from the 1800s. And I was looking for somebody, I can't remember the surname, but it began with L, and my surname is Lowe. And then I saw a Lowe, well, two Lowes. I think there was T.R. Lowe, and then there was an M. Lowe. And I, just curiosity, I looked at both, and then I thought, oh, I'm actually there. And what is really weird is seeing yourself there with the year date, your date of birth, your year of birth, although I must say, when you look for it, actually, it's a couple of years out, so I've got to correct that. But then you see a little dash, because I'm used to seeing, you know, the birth and the death of people. So that is kind of weird. And then there were lots of other pictures that I was unaware that were stored in the archives of when I first started as well, and a little biog about me. So that little girl in the middle is me, and she's been able to tell her story so that in many decades, hundreds of years to come, that people will be able to find me and know all about me and my contribution to science, which is great. This black and white picture here is actually when I first started at the museum, and it's in a kind of roughly scientific paper about trolleys and the safety of carrying museum specimens. And I didn't, and this is where, you know, like Leanne pointed out, there's not enough of me in marine science or in the natural history world. And so when I first started at the museum way back when in 1991, I think this is 92 or 93, I was the only young female of colour in the museum, and they wanted a young person to model with the trolleys. And I don't work on fish, I work on, you know, crabs, lobsters and corals. And so that's how I got into that paper, because they wanted a young person. And then the one at the end is me in a pose of somebody who I really admire, another female scientist who used to work on crustacean like I do at the museum, a lady who was from Scotland. Her name was Isabella Gordon. And so I use a lot of her tools and equipment as well. And so I was doing a similar pose that she did, I think, in the 1930s. So that's what I mean. So a lot of what I'm going to talk to you about this morning is linked to this document. So I'll read this out. 2007, I'll read it out here. 2007 marked the British bicentennial of the abolition of the transatlantic slave trade. The Natural History Museum contributed a unique angle to the nationwide dialogue on the transatlantic slave trade. The museum itself was founded within the context of empire, colonialization, and exploration, and as such, there are many connections between the transatlantic slave trade and its history. So that is the front end of what turned out to be a document with 10 chapters, documenting and kind of hinting on stories of various people, but in there were some stories which I have developed, which I will tell you now, of people of colour, their contribution to science and the natural history world. I would say that document is now in its entirety since October last year, back on the museum website. I pushed for that literally for 10 years to get that together. What had happened, and this happens with a lot of projects, so it's not just about this topic. We've all got to sort of think about, when we do projects, you get funded for, you know, a finite period of time. You produce documents, and as wonderful as the internet is, it can be very chaotic as well. So when I went to look again after you know, all of this bicentenary was over, went to look for this document because of some of the stories that initially had resonated with me, I only found one chapter of the book, or of the report. And so it's taken a while, and because of staff changes, our community engagement team got disbanded and all of that, who were instrumental with this, because at the time, and this is another thing to be aware of, I'm aware that when I am rolled out as a person of colour for particular events, but I do think that if it's not me, who else is it going to be? So, you know, I was quite, you know, a big part of these events at the museum. So essentially these documents are there, and they are wonderful documents. It's a real shame that they weren't there in their entirety, you know, from day one right up to now, because the response on Twitter has been amazing, and people saying, oh, this is like great information here and everything. And now that decolonising spaces, I mean, you're decolonising near enough anything, so we're talking about decolonising natural history and narratives and diverse voices being heard. This is key to that moving forward, and it's nothing for the museum to be ashamed of. It's to be celebrated, basically. So, I don't know how many of you have noticed, when you walk through the museum central hall, Hinsey Hall, there is a wonderful ceiling of over 160 panels, botanical panels, you know, images of plants that were instrumental to the British Empire. Tea, coffee, cotton, you know, all of those products that were taken, sourced from the empire due to colonialisation of various countries such as Jamaica, and it was also at the time when it was the slave trade. So, one of the stories that was in that document, Slavery in the Natural World, was about this plant and the person connected to this plant. So this ceiling panel of Quasi-Amara is actually, when you go to the up on the second floor of the museum, and you're facing the giant sequoia, right on, so it will be on my left-hand side. If you look above, diagonally up above the sequoia, you will see the ceiling panel. And this panel, the plant in this panel, so I've got a picture of the actual plant here, was named after this gentleman, Graman Quasi. And it was Carl Linnaeus, who Leanne sort of spoke about, and we're in the Linnean Society here now. He named this plant after Quasi. Now, how that came about, Quasi was, as a child, he was born in Ghana, but as a child, he then, due to slavery, he was then moved to Suriname. And that was a Dutch colony at the time, in the early 1700s. He moved there, but he then, as he grew up, he had an interest in plants and their medicinal properties. So he then became he established himself as a botanist and a healer. He found out that there were medicinal properties in Quasi-Amara that could actually help with intestinal parasites, help reduce fever and things like that. So where he was within that community, within Suriname, kind of, you know, held in high esteem, there was another sort of side to the story which the indigenous peoples didn't quite like, that he became a scout for the Dutch colonialists. So he also lost his ear in fighting, let me get this right, the Maroons as well. So it was a little bit divisive. But going back to the plant Quasi-Amara, that plant then, because Quasi was friendly with the Dutch colonialists, he also came across a Swedish plantation owner called Carl Gustaf Dahlberg. And that plant and the information got into the hands of Carl, who then in 1756 came back to Europe, and that plant was transported and put into the hands of Carl Linnaeus. And what I'm trying to say here is this story is not embedded in any of the text at the Natural History Museum at the moment, and there was a refurbishment of those galleries in 2017. And so I think that how wonderful it would be to celebrate that through Quasi, that this plant was named after him by the grandfather, the godfather of taxonomy, Carl Linnaeus. But it does pull up those issues that at that time, a lot of enslaved people, their local knowledge was taken away from them and brought to Europe and commercialised. So, for instance, for Carl Linnaeus, he then published published a paper, a scientific paper on that plant, and it went to the wider scientific community. And actually, you know, it formed a bitter tea. It's used in pills now. So it's actually sort of used in medicine today in the wider world. So this is luckily one way that a person of colour has actually been acknowledged for their own knowledge at that time, but not a lot of people know that. So by me talking about it, the document that is now on the museum website, we're spreading that kind of knowledge. It's a celebration as well. And it's also, hopefully, eventually, showing people of colour that there are stories in these cultural places for you that we have also contributed, and it's something for everybody to know about and to celebrate. So what I'm showing you here is a collection that I actually look after, and it's this man here, Charles Darwin. These are some of his barnacles, so I actually look after his barnacles. And I know you all love barnacles. Okay? So this is the most glamorous you're going to get. Stalked barnacles here. But why I'm mentioning this man, and again, Leanne, I saw your blog as well. I was really pleased about it as well. So spreading the word. Leanne had mentioned this chap. So this is John Edmonston, and he's the man who taught Charles Darwin taxidermy. Now, again, that was a small snippet of the story within slavery in the natural world. So John Edmonston was, how he came across Charles Darwin. He was a freed Guyanese enslaved person who had a master called, I'm trying to get the name right now, I've forgotten the name completely. But anyway, I've forgotten the name. But anyway, his master knew a naturalist called Charles Waterton, who is a naturalist who resided and lived in Wakefield. And John was actually taught taxidermy by Charles Waterton. And you put it in your blog as well, Leanne. Charles wasn't very complimentary about the skills or teaching John, but eventually John ended up in Edinburgh. And how I found out a little bit more about this story was because I was doing some research into those barnacles, I still am, and looking at the Darwin online, the correspondence online. And the only way that you can tell that John Edmonston was a person of colour, the term that Charles Darwin uses is in a letter in 1823, he writes to his sister, Suzanne. He mentions that, I've come across a blackamoor that can teach me taxidermy, and he's at a really good rate, and I'm going to have a couple of weeks training from him. And that's the only way. And so again, this is the thing that a lot of people of colour at that time, they weren't telling their own story. It was being written for them or about them. And so, as Leanne said, we currently don't, the Linnean Society doesn't keep those, those kind of ethnicities, that kind of data. But, there's something to be said. I mean, we've got the internet now, we've got lots of digital, we've got the images so people will be able to see what I look like. But, it's really hard when there was, just letter writing at that time to kind of find out who were the people of colour at that time. So it's really great to be talking and celebrating, again, John Edmonston as well, that his contribution to science, teaching what we hold up, as a great European scientist, Charles Darwin, that, before he even went on the Beagle, that he was being taught taxidermy and thinking about, those skills he used while he was on the Beagle. He would have heard all those stories about South America from John as well. And that must have, inspired his mind, intrigued him, maybe informed him in many, many ways of what he ought to look out for or what it was going to be like on his journey on the Beagle. So then we come to Sir Hans Sloane. For those of you that don't know him, his collections are the founding collections of the British Museum. The natural history collections were based there, but when that collection grew too big, then the Natural History Museum, where I work, was actually built to house those collections. So Hans Sloane went collecting at the age of 27. He went to Jamaica. He actually, throughout his expeditions there, collected over, 800 plants. And what you can see there, the plant there is the cocoa. And he's associated with Sloane's chocolate and, and the cocoa, the bitter cocoa being commercialized into the chocolate that we eat today that's added sugar. But at that time, when he was there in Jamaica, he was also, and this is the thing, there are other sides to any story. So as well as collecting, he was also, well, one would say, he definitely had enslaved Africans collecting for him, in the Caribbean. So, and so his collecting and the time, and he was documenting about the slave trade. And those stories are not always teased out of the time when people were collecting. It was a very tragic time for people of colour. So he was using a lot of their local knowledge of where to collect certain plants. And I would say he wasn't very complimentary in terms of the uses that people of colour had, the medicinal uses. He didn't think that they could use them properly without wider knowledge. It's these stories that need to be told to inform people of the time, and that whereas, Sir Hans Sloane can be celebrated for collecting, and I perhaps would not be working at the Natural History Museum if it wasn't for some of his founding collections. There is something to be said that, and to acknowledge that Hans Sloane also, he married Elizabeth Rose, who was a plantation owner, and would have had slaves, and is very much linked to the sugar trade as well. So a lot of these collections have been collected off the back of the slave trade. That's something to be acknowledged and for us to move forward with, but to tell those stories again, because if museums and cultural spaces want more diverse people, whether it's in terms of gender or ethnicity, people have to know that organisations are open and telling all the stories as much as they can do, and being to tell the truth. So we're going back to this document here. So that's just a snippet of some of those stories that I told you. But then, literally 10 years in the making, I wanted to do a guided tour about all of this. And because of new members of staff that had arrived at the museum, I bent their ear a little bit. And October last year, I was able to do my first Black History tour within the Natural History Museum, within the Hinsey Hall, taking people around. So I've only done the tour twice, October last year, so it was part of a lates event. So a lot of organisations and museums have lates the last Friday of the month. So it was October lates and November lates that I did this Black History tour. And literally I couldn't believe it, because we didn't do much advertising about it for the October lates, but there were about 18 to 20 people that turned up this tour. And I said, "Are you really here for this tour?" I said, "Do you know how excited I am? And we're actually making history here." And they were just loving it. And I wouldn't even say, it wasn't just people of colour that turned up, because this kind of information is for everyone, to share with everyone. And so this is just a tweet from a chap who was a school teacher, and he was so excited. He said, do you do this all the time? I said, "No, this is the first time." He said, "Oh, do you do this in schools?" "No, not yet." But what I do on the tour is that I take people, so you can see one of the pictures here, well, two. So there's a sequoia on your right, and then there's Charles Darwin's statue at the top of the main staircase in Hinsey Hall. So I talk about John Edmiston at the foot of Charles Darwin there. I'm talking about Kwasi and Kwasi Armara specimen, taking people up to the second floor to look at the ceiling panels. And what that also gives, because a lot of people said, oh, I've been here 101 times, and I did sometimes sort of think, what was the other story behind the ceiling panels or whatever, those other kind of stories. And they absolutely, as you can see here, this guy says, a brilliant tour exploring a different side to natural history that is often not talked about. Really made me think about the untold stories and the contributions that have been erased by colonialism. Would love to come to the next one. So, people actually really want these stories, and they want to see themselves in the galleries. We have to next look at the interpretation, how we write the text amongst some of those specimens, and to bring out more specimens if we can do, or do more tours like this. So I just want to leave you with two things, really. So this is a quote that I've used quite a bit. So Monica Montgomery is a co-founder of an organisation in the US called Museum Hue, and it's basically a group of people of colour that get together and look at issues such as lack of diversity in museums. So it says, it is uncomfortable for many institutions to even broach the subject of the museum's complicated relationship with audiences of colour. But Black Panther, the film, has created an impeccable opportunity for institutions to begin a dialogue with their community. And that's what we want. It's the start of a dialogue, decolonialising various spaces. It's that dialogue that we need to have. And this is what I want to leave you with. Thank you.
Okay, thank you. Okay, I think I'll switch this on, yeah. So first of all, I'd like to say thank you to the Linnean Society for inviting me here to talk today. It's the first opportunity I've had to share the results of this research that I've been working on, sort of on and off, stop-start basis over the last 3 years or so. In the next half hour, I'm going to show you some of the amazing images that I've had the privilege of working quite closely with, and tell you the story that's contained within their making. So the natural history drawings referred to in the title belong to the genre company drawings. So I thought a good place to start was just to talk a little bit about what company drawings actually are. So they're artworks made in the late 18th and 19th centuries. They had Western commissioners from local artists working in South Asia and Southeast Asia. And the reason there were quite so many Western commissioners in those areas is because they were working for the British East India Company. And that's the logo of the company up there. So these works became known generally as company drawings because of the association. And they're generally understood as hybrid works, which means that they were made on Western paper. So you can see that kind of watermark. They've got pencil underdrawing. And then an outline was applied in waterproof ink. In the case of the South Asian works, that was probably with a squirrel hairbrush, or possibly with a squirrel hairbrush. And then they were painted. And there's a really quite diverse range of subject matter within natural history itself. It could be insects, plants, reptiles, fish, animals, birds. And then otherwise, there's quite a lot of portraits, architecture. And these two pieces came from completely separate albums, but they were recording the occupations of Indians at that time. And I think they offer a really quite interesting snapshot of Indian artists working in the early part of the 19th century. So this one, dating from 1815, is the earlier one, and I've enlarged his materials. And I think this looks like a very traditional set of materials. He's got those pigments in little individual receptacles, which look very much like mussel shells. And then in this one from 10 years later in 1825, he's got himself a very nice imported kit, looks like. And I think the similarity between the layout of his box and this one here is really quite incredible. This one is in the Museum of London, and it belonged to Isaac Smith, who was the nephew of Captain James Cook, and it was used during the first voyage of the Resolution between 1772 to 1775. These mussel shells are actually from a class that I attended last year at the Prince's School of Traditional Arts and Crafts to get some hands-on experience of Indian painting techniques. So these were used by another student on the course, so it was just quite nice to see that the shells are very much still part of that practice. So the drawings that this study that I've been working on focused on are those commissioned by Francis Buchanan-Hamilton. They were made during the first ever survey of the Kathmandu Valley. That was done between 1802, from 1802 to 1803. And there are actually no images of Francis Buchanan-Hamilton, so I have chosen to represent him through some pages from his lecture notes made at the Royal Botanic Garden in Edinburgh in the 1780s. And in these lectures, he would have been introduced to the cutting-edge science of the time, which was this classification system introduced by Linnaeus. And in his future career with the East India Company, he had plenty of opportunity to put this into practice because he trained in medicine at Edinburgh, and botany was very much still part of the medical curriculum at the time. And he was employed by the East India Company in the Bengal Medical Service from 1794, but his skills as a naturalist were highly valued by the company, and he was sent on various surveys. So between 1794 and his retirement in 1815, he conducted, first of all, a survey of what's now Myanmar, and then Bangladesh, and then he was sent right down to the south of India, to Mysore. And then just getting back from Mysore to Calcutta, he was told that he only had a week to turn around and prepare himself to head north and join a diplomatic mission that was due to enter Nepal. So he spent just over a year in Nepal, then came back, and then spent basically the rest of his career involved in a survey of Bengal. So the work that he did in Nepal, he had a real hope of publishing the first ever flora of Nepal. And sadly, that did not actually happen. Hardly any of his work done in Nepal ended up being published. But the pioneering work that he did there has led to him being known as the father of Nepalese botany now. So the reason for making illustrations like this one at the time was really just because, well, there are no cameras or anything. And although he was making notes and collecting herbarium specimens, the herbarium specimens contain a lot of information, but unless you're used to looking at them, they don't really look very much like this. I know because actually I work as a botanical illustrator at the Botanics in Edinburgh, and I spend a lot of my time working from herbarium specimens, and I'm trying to turn them back into this. This one here is on display upstairs in the library, so you might want to go and look at it, and it has the corresponding herbarium specimen, so you can see what these look like. So talking about people being hidden through this history, sadly the artists' names are very, very rarely recorded, so these collections of works tend to become known by the name of the commissioner. And it was very common practice at the time to have copies made for distribution, dissemination of knowledge. So Buchanan-Hamilton chose to have 2 further sets made of these drawings, and he gave one to Richard Wellesley, who was the Governor-General of India at the time from 1798 to 1805, and also a friend and naturalist, John Fleming. So the Wellesley collection, you can see, have been bound into a volume, and they are currently within the British Library collection. And then the Fleming works, the Nepalese works, are now distributed amongst over 1,000 works that make up the Fleming collection. So, and they have been individually trimmed and window mounted. And the Buchanan-Hamilton collection here at the Linnean Society came to be here because he was hoping to publish these works, and realizing that the workload with the East India Company, meaning that he would have very little opportunity to do so. A friend of his from university days at Edinburgh was James Smith, the founder of the society. So he gave his collection of drawings and his notes, and he had expectations that Smith would assist in the publication here. Sadly, that didn't happen either. A few, I think 37 botanical descriptions unillustrated were eventually published, and only 12 of the illustrations were ever engraved and published. So what I'm hoping, as I've been talking, is that you've been looking at these drawings and are as amazed as I was when I first saw them at just how exactly similar they are. Same format, quite complex root systems are exactly the same, the arrangement of the flowers. So it just says to me that there had to be a copying system on the go, some method of copying them. So, yes, one of the questions is how were these made? So you can see a little bit of oxidation on the inflorescence here in the Wellesley and also the root at the Natural History Museum. That's degradation of lead white, but otherwise really quite good condition and very similar. But an important difference in the Buchanan-Hamilton collection is that they can appear unfinished. Areas of them aren't fully painted. So a couple of reasons that might have been is were they the originals by the artist who accompanied him to the Kathmandu Valley in Nepal, or were they a very late set of copies made and unfinished when Buchanan-Hamilton found himself the opportunity to return to Britain and the intention of giving them to Smith and hoping to have them published? So having looked at the drawings, the three research questions I set were, which are the originals, what was the copying method, and actually can the order the copies were made in be told? It's a very, very low-tech study because it was just me going to archives and having a look, basically. So the most equipment I had was a torch. So I was looking for clues such as the watermarks, which are not readily visible, but you can see when you shine a torch through. And then a really interesting feature of all three sets of these drawings are these pinholes, which, sorry, trying to find the pointer. These are not to scale and not where they are on this drawing, actually, but I've just tried to indicate where you might find them on the drawings. They were actually really quite difficult to see. This is one enlarged. So they are pairs of pinholes, and you can see here where the pin has been pushed through and has come out. They're only about 5 to 10 mils apart, so really a bit like looking for a needle in a haystack on some of them, and you really do need to use a torch to try and work out if it's just a mark on the paper or actually a hole. And then the other thing was just actually looking closely, very closely at the drawings, seeing if there was any sort of handprint of particular artists or similarities and differences. So first of all, comparing paper types and watermark. So there are a couple of paper types. There's laid and wove. Laid was, by this point, actually really quite old-fashioned. These grid lines come from the mold that the paper was made in. And in the 1750s, the Whatmans invented what's called wove paper, where they were using a fine mesh on the mold, and it eliminated this kind of grid patterning. By the 1780s, everybody was getting in on the act of making this wove paper. So I don't actually have a photograph of an E&P, it stands for Edmead and Pine, but the date of the paper was 1801. So to keep a track of what was going on, this is an excerpt from the type of tables that I drew up. So the first column is the Buchanan-Hamilton, then the Wellesley, and then the Fleming collection. The sheets of paper themselves had been halved, so there's 2 drawings per sheet, which means that there isn't always a watermark on every piece of paper that you look at. Although the E&P and Whatman were both wove paper, there's enough difference just visually. The E&P was a lighter color, lighter weight, and when you shine a torch through, a much more easily seen grid. So although there isn't the watermark, I've occasionally written seems like E&P. It doesn't seem like the Whatman paper to me. So from this, all of the Wellesley collection are on the Whatman paper. A couple of Buchanan-Hamilton only on the Taylor Laid, and the Fleming are kind of 50-50 between the E&P and the Whatman. So the Wellesley and Buchanan-Hamilton from this have the most similarity. So then looking at the pinhole positions, to do this, I drew up, and 1 centimetre square grid all over an A2 sheet of paper and had numbers coming down the side and letters across the top. To position them on the drawings, I was looking for obvious points like down here or here. I've used, I'd say third leaf up on the left, and this tip would be at 24E. So I'd select 2 or 3 points on the drawing itself so that I could reposition the grid and then look for the pinholes. And I also, as well as mapping their position, I measured the length of them and also whether they were horizontal or diagonal or vertical. And what I found from that was, well, the other thing I should say, there are 122 species drawn through during the year in Nepal. So to keep the study on a sort of manageable level, 25 of these images were chosen to focus on in particular. So I do have data relating to all 25, but also because of the way that the collections have been housed in the 3 different institutions, they tend to come up in different orders. So it's actually, you end up not getting all the data on each trip as you're looking at different ones. So out of that 25, I have complete data from all 3 images in the sets for 14, and out of that 14, 2 of them said that there was no data available for the Fleming because they've actually been trimmed and mounted, so the places where the pinholes were have been cut off. Not hugely helpful. And of the 14, 7 were really exact matches, like this sheet here. This shows really clear correspondence in length, direction, position between the Buchanan-Hamilton and the Wellesley, and then the Wellesley to the Fleming. So, but something that came up was this fact that the Wellesley very often had 4 sets of pinholes and the other 2 would have 2 sets of pinholes. So from this, the Wellesley generally matched the Buchanan-Hamilton and the Wellesley generally matched the Fleming. And so this is the number of pinholes table, so you can see quite clearly that 11 of the Wellesley have the 3 to 4 sets. So anything more than the two sets per sheet. 3 of the Buchanan-Hamilton have the 3 to 4 sets and only 1 of the Fleming. And so the story so far is that at the moment the Wellesley are coming out as the contenders for the originals because they have the most consistent paper stock, they're all full colour, they're bound neatly into a volume, and these pinholes actually show a very clear correspondence that the Wellesley sits between the other 2 collections. However, the other thing that I was looking at was this close examination of the works themselves. And I've gone back to have a quick look at this image again because when you look at these completely finished ones, there's actually no trace of the working method left at all. So it's just, they've just landed on these bits of paper and you can't really tell very much, which is where the Buchanan-Hamilton collection here was just, a real source of knowledge and finding out about the working methods. So focusing on this leaf here and an area of the flowers up here, I'm hoping that you can see this because I've tried to darken down the underdrawing. You can quite clearly, I hope anyway, see the underdrawing. So this leaf has changed position a few times, a bit of hesitation about where the veining goes. And then this is a complicated inflorescence, and you can really see this person thinking about what they're doing here. They've been working it out. And I was also thinking that a lot of people think when you're drawing plants, they remain static, but actually they don't. Through the day, they'll follow the light. And then you've got buds here and open flowers, and something you've been looking at will open as you're drawing it. So, there's been a lot of thinking and understanding going on here. And to me, that just says that these are the originals. They must be, because they have that level of thinking about it. It's like the first time you've seen something and you are working it out. And if you want a closer look at that, you can come up and see if you can see that on the original here. But actually, I came across another piece of evidence which points to these unfinished works being the originals. And we're going to go on a short jaunt to India for a few slides and a change in subject matter. I had got funding to go and just as a part of the kind of wider building a picture around this subject. So I managed to go to the Acharya Jagadish Chandra Bose Botanic Garden, formerly the Calcutta Botanic Garden. And they have this amazing, collection of the Roxburgh iconies, which I don't know if you're familiar with. So there's over 2,500 of these drawings. And yeah, they're housed in these big red volumes in this cabinet. So that was really interesting to see, but the clue came when I visited the Asiatic Society. So this is sadly this building here with the name on it, and this is as far as cameras and computers can get to the Asiatic Society, so it was just notebooks and pencils inside. And I spent the whole time thinking, I wish I had a camera, because this is the kind of image that I was seeing in there, these amazing bird drawings which come from the Survey of Bengal, which was after the Nepal trip. The ones in the Asiatic Society weren't, this one's from the British Library, so the company doing things in duplicate or triplicate as usual had sent these back to London. But the ones in the Asiatic Society had actually quite a lot of degrading around the edges of the paper. So to conserve them, they'd been pasted onto larger sheets of paper, and then quite an elaborate window mount was pasted over as well to prevent the surface of the drawing from being transferred onto the next sheet. And I'm describing that because there was something, they were in 3 large volumes, and something that happened when I was looking through, I could understand this care taken for the birds, but very occasionally, there was just a blank sheet of paper, treated exactly the same way. But then, looking through them, I don't know if you, can you actually see that at all? I tried to mock up a ghost image, which was in reverse. And I suddenly realized that actually these blank sheets had been cover sheets separating batches of the drawings. And then the real clue came here with these words, originals wanting, and this was in Nathaniel Wallace's handwriting. So he was the next superintendent after Roxburgh of the Botanic Garden from 1817. And all the drawings after the words originals wanting were full colour. Because a feature of the ones in the Asiatic Society, all the ones in the British Library are full colour, but in the Asiatic Society, they were very much as these drawings are. So they were partly filled in, fully drawn, but there was, the beak would be filled in, some of the scales, examples of the feathers that could be worked up later. So with these 2 bits of evidence, I think we can say that these Buchanan-Hamiltons are the original drawings that were made by the artist who went with Buchanan-Hamilton to Kathmandu. And then, yes, another thinking about these being the originals, so how were the copies then made? And a clue came. This is the Quercus that's here at the Linnean Society, but when I was looking through drawings of the Fleming collection in the Natural History Museum, just by chance happened to notice that on the back of one of those drawings was a complete drawing of this image, which had been partially rubbed out. And I have tried again to show you what some of this drawing looked like. So focusing in on the acorns, I think you can see here that this is very, very close match, but also a very different type of drawing. It's just very easy. It's just one confident line. There's been no hesitation, no rethinking, or trying to understand. So that just says it's tracing. It has been traced somehow or other. So how would you trace? I think I read that Canson claimed to have invented commercially available tracing paper in 1810, but before that, so various methods that I've read about are squaring up. And given that you can see some of the underdrawing traces still on these, and you would have to draw a grid on all 3 sets to square up, and there's absolutely no evidence of that. I don't think that's right. Pouncing was a technique by which small holes were made, pinpricks, and then something like charcoal dust spread over, and then you literally join the dots underneath, but there's absolutely no holes in any of these. Transparent vellum is something that I came across, very particularly fine grade of vellum that you could see through. And also making tracing paper using a thin weight of paper and then a mix of linseed oil with turpentine and left to dry, which if you have any knowledge of art materials, using something oily with something water-based, these works are all water-based, that just wouldn't work at all. It would cause the paper to resist. So yeah, so, and then the other clue, this is a picture of Nathaniel Wallach, and it's actually an image that is here at the Linnean Society, and you can see it on the way up to the library. And I thought it was about time that we introduced him a bit more properly, because his evidence has actually proved quite, I think, literally illuminating in this case. So there is this quote here, which is in a book called The European Discovery of the Indian Flora. It was written by Ray Desmond and published in 1992. And he says that Nathaniel Wallach was describing the copying of drawings at the Botanic Garden by the aid of glass panes. Really, when I started thinking about this, the only thing I could think that glass is useful for is the transmission of light. So is he referring to some kind of light box? Very frustratingly, this quote is not actually referenced. So hadn't actually found the source of this quote and seeing if there was any other further information around it. So, but thinking about using tracing paper or the light box, if it was tracing paper, you wouldn't actually need to pin these sheets together, whereas you would if you were using a light box, because you're just transferring from the drawing to the other sheet of paper. And I also actually started making a lot of sense of things that I was finding. So going back to this collection again, this is one of the 7 out of the 14 where I had mostly exact matches, but then the very odd one that just wouldn't match up. And I thought I'd maybe made a mistake of reading the grid or something like that. So I went back and remeasured everything. And the note I made to myself was everything on the top is exact for all 3 sets. There's something happening in the roots here. And this is the results from the table. So you can see the discrepancy comes between the Wellesley and the Fleming collection. And you can see that that's the pages out of the book. So this is the Wellesley collection, and this is the Fleming one. And what it actually comes down to, these don't show it hugely well, is that actually this tip of the root here is shorter than it is up here. And that was enough to push it off on the grid. And when I started looking through the images again, seeing which ones were mismatches, the thing they all had in common was that they were complex pale-toned areas that had the mismatch, or areas like this, which were unfinished on the originals. And so this is the one here at the Buchanan-Hamilton collection, and this is the Wellesley version. And one of the very, very, very few occasions where I came across any underdrawing on any of the other collections, as in kind of that thinking underdrawing that the originals have, was in this area of the Wellesley collection. I don't know if you can see it there, but there's been a few attempts at finding the exact position of this leaf. And if you actually map these leaves here and under here, you'll find there's definitely one missing, and this one has slightly changed position. So these have been done by eye. It just, if there's the light transmission through the 2 sheets of paper hasn't been enough to allow for straight copying. And this also made sense of this light anomaly that was going on here. This is one of the rare full colour images within the Buchanan-Hamilton collection. And when I mapped the pinholes, rather than having the Wellesley collection as a go-between, this Buchanan-Hamilton one actually has pinholes which relate to the Wellesley and to the Fleming. And the Wellesley collection in this case is not the go-between. So where there is a full colour image available, copies could be made straight. And going back to this one, where there was only a partial image, it makes complete sense for you to then work up the full colour image from which to make further copies, because that would be easier. So, yeah, superficially, the Wellesley and Fleming seem more complete, but this one here is, you can see here that there's this fruit that's been added to this drawing, and when it comes to the copying, there's a pencil outline in the Wellesley, and by the time it gets to the Fleming, it's just gone. It hasn't made it through the process. So this, again, is a kind of complex pale-toned area. So the outline has been transferred here, but that would have required further work by eye, so actually looking and transferring that information. And for some reason, whether time or whatever, it just hasn't occurred. And as I say, by the time you get to the Fleming collection, it's just gone completely. But that's a very obvious loss of detail. But actually something else that has happened through this copying process is this is from the, it's this drawing at the Linnean Society, and just looking at it, you can see that this has been drawn from life, somebody observing closely from life. So there's the inclusion of these little hairs, the scale under the bud, little hairs around this bud and on the leaf attachment. And these have just completely gone. So although there's extra veining and fully finished to a certain extent, the other twigs, actually there's quite a loss of information. And then by the time you get to the Fleming collection, there's just very little of this really nicely, closely observed from life detail left. And that's something, it is a feature of so many, well, all of the drawings. The Buchanan-Hamilton, although seeming less finished, the reason these leaves aren't painted in is because there's no information from the originals. And with this one as well, these are just wash, so that's all that could be done in the copies. And in this one as well, you've got, that's just actually a really nicely observed way that the structure comes up and opens out into the leaf. There's little white dots on the bark, and these flowers are really quite complex with all the little stamens shown. So this has been transferred really quite well into the Wellesley collection, but things like the way that where the veins fade off towards the edge, that's again something that you would see in life, which has not been transferred so well. And then actually something that has occurred to me, when I was looking for the watermarks in the, between the collections, something I did notice in the Fleming collection was a lack of lead white, which shows up as really opaque on the other collections. And that's not because it's not consistent all the way through, because the orchid that we looked at first had some oxidation on the roots. So some of them have the lead white, but some of them don't. So I'm actually wondering if this person was being encouraged to work in a more Western way with more watercolor-based paint rather than the gouache, which requires a completely different approach. Here you can fill in dark areas and then just put light over the top, but watercolor is the absolute opposite way around. You have to leave the lighter areas. And this person, I think that's what's happened here, and it is actually quite a struggle to do that when you're painting leaves with watercolor. So just to finish and summarize, I think there's no doubt that these are the originals by the artist who went with Buchanan-Hamilton to Nepal. I do think that the copying method is tracing over some kind of light box, but when I first had this vision of light box in my head, I'm thinking about the ones that we plug in today with really bright lights. However, if you kind of change your thinking on what a light box could be, it could be a window. And something that, if you do look more closely at these images, you'll see is actually this is very, very lightweight paper. It's much more lightweight than we would use for watercolor today. And I have actually tried with, these are about 90 grams weight in paper, and I have used 90 pounds paper, and I have tried it with 140 pounds paper, watercolor paper today, and just held up against the light, and you can actually see quite a lot through just from a window. It does explain this position of the Wellesley collection as a central sort of information transfer between them. And I think from really sort of quite a small scale and relatively low-tech study, it's started to build up a picture of there's a single artist out in the field working with Buchanan-Hamilton. And then I think he may have had something to do with the Wellesley collection copies because they are quite high quality with more retention of the information. But I do think that the Fleming collection have maybe been set up, a workshop set up, because there is clearly more than one hand going through that collection if you look at them. Some of them are actually very high quality, and actually they tend to be the ones with more traditional materials, I think, in the use of lead white And then the slightly looser ones are the ones that I suspect are maybe more watercolor-based imports, possibly. So that's as far as I've got, really. It would be nice to be able to tell you more definitely who the artists were, but that really wasn't recorded. But there is Dr. Henry Nolte, who did work at, he's now retired, he worked at the Botanic Garden in Edinburgh. He spent over 25 years specialising in looking at this kind of work. So I think we're getting quite close to maybe establishing some names for the artists, which would be very nice. And for myself, I'm hoping this isn't the end. I would really like to take this forward. My particular interest is, as a botanical artist, I trained in fine art and I'm self-taught as a botanical artist. So what I've always thought when I looked at these drawings is that there's just so much information that these people have brought to this work themselves. And I really want to examine them from all sides. So how these people adapted their work towards the scientific requirements and what things the Westerners might not have understood about their practice methods. I mean, there's a lovely reference with Buchanan-Hamilton slightly complaining about how long his artist is taking, but so yeah, around that kind of thing. So hopefully you might hear more from me. Anyway, thank you very much for listening.
Okay, thanks a lot for the invite. I'm really happy to be here. So basically, I'm going to tell you the story of a research project that we are launching that is based on ethnographic research we have done in Mexico. So I'm going to tell you a little bit about that. It's called The Frog and the Vine because we are following two organisms. One is the poison dart frog that comes from Colombia in the Chocó, which is an indigenous region. And the other is ayahuasca, which is, probably you know about ayahuasca, I don't even have to introduce that. It's quite a popular vine. And basically these two organisms have been at the center of lots of biopiracy issues in Latin America especially. And so I'm going to tell you a little bit where I come from. So I come from doing this sort of thing, which is anthropology of science. So I do ethnographies of scientists doing their work. And my job has been to follow scientists studying genomics for around a decade. And what I do, I'm Mexican, by the way, if you were wondering, what I do is that I was obsessed with this notion that we Mexicans produce called genomic sovereignty. So we thought that our genetic background was unique, which is a weird thing. And that we needed to defend that from foreign interests, basically the US, that would patent our genome and make medicines out of it. And I mean, this is based on hundreds of years of biopiracy scandals in Mexico. Do you know the poinsettias? Yes? That's an insult for us. Poinsettia was the ambassador of the US that took the Nochebuena, which is an endemic plant in Mexico, and then patented in the US as if it was his great thing. And I could give you lots of examples of that. So we Mexicans are really kind of like angry with this sort of notion of how history is rewritten once plants and animals travel around. But we are not the only ones. So this is a Latin American thing. And it comes from a lot of indigenous activism. But I was amazed with what was going on in Mexico, because in 2007, when I started doing this ethnographic work, as I told you, we created this notion of genomic sovereignty, and what that implied was that we created laws to protect our Mexican genome. It was not just a notion. So we spent years discussing in the Congress about what it meant to create a framework for genomic sovereignty. And basically, what I'm going to tell you about is derived from those 2 years that I spent with genomic scientists in Mexico. So you know, when you're doing ethnography, you take like a long time to really get intimate with the people you're working with. And they were really teaching me. So you see those images, that's the lab. This is how, all right, sorry. This is how it was presented publicly, and this is actually what we did during those 2 years. And this is just to give you some background of what is the methodology that I generally use to talk about science and engage critically with scientific objects. It's the other way. And basically, what we are trying to think is, how can we produce an indigenous bioscience? I mean, the idea is that science and indigenous knowledge and cosmologies are kind of antithetical, right? So you have this cosmology that is based on tradition. Some scientists would say it's superstition, because they have this colonized notion of what is real knowledge. And then you have all this indigenous knowledge that when it travels, turns out to be an anecdote. And then it's patented somewhere else. So there's a real problem when we think about these two traditions, this modern Western tradition about what counts as science, and this other cosmology of indigenous people. And if you think that it's just a question about recognition, we think it's much more problematic than that. And when you think about the animals, then a beautiful example is that when you speak with some of the indigenous shamans, they tell you that they knew about ayahuasca because the spirits revealed that to them. And this is something very interesting. It's a radically different ontology when it comes to thinking about science. And what has happened so far is that scientists try to select, pick and choose what bits of this indigenous knowledge fit their narrative. And they say, well, of course, the most, I think, progressive and open-minded of those think like, oh, well, indigenous people knew what science is now proving, right? But you already see the hierarchy here. Science proves what indigenous knowledge might have just guessed or hinted at. So can we really produce an indigenous bioscience, or this is impossible? And this is a question that matters for the issue of biopiracy for two reasons. One is because patents in the global north are basically based on the contribution of knowledge. How you prove that someone or some group contributed clear knowledge to whatever you're patenting, the organism, the process. And secondly, because the two objects that I'm going to be looking at have already been subjects to patents. It's not that I'm a hypothetical thing. This poison dart frog was patented by John Daly, who was a medic, following these beautiful frogs from Ecuador to Colombia. And he found out that one of those frogs called Phyllobates terribilis, the poison dart frog, is the most toxic vertebrate on Earth. And he was hoping to produce a medicine, I'm going to tell you a bit more about these cases, that would substitute all opioids. So it was like wow, it was the transformation of medicine. And then you have ayahuasca, and ayahuasca is a more interesting historical object in the sense that it has been around for hundreds of years. It was also patented in the 90s with all these bioprospection craze. There were lots of scientists outside their laboratories finding these organisms and trying to extract chemicals out of it. And I want you to go with me in a little trip that is going to take us from the Mexican case that I spent 2 years researching into the cases of ayahuasca and the poison dart frog, and then to the contemporary issues of biopiracy for the Colombians and the Ecuadorians, which are the two communities we're working with. So product of what we were doing in Mexico City, I'm part of this network of indigenous scientists that want to produce a different sort of engagement between Western biomedicine and indigenous knowledge. And this is where it comes from. So we are trying to deal with this issue, not only as a question of inclusion in science, but also as a question of transforming the way science and indigenous knowledge operate, and the hierarchies that are kind of constructed within these relationships. So let me take you to Mexico. So when I started this ethnography, Mexico had invested $120 million of their health funding to create the most ambitious health genomic center in the global south. And with it, it produced a policy agenda that was taken up by China, Brazil, India. The global south was really excited about not only the investment, but the ideas that came with the Genomic Institute of Medicine in Mexico. As you can imagine, it was a complex process. There were corruption scandals. I can tell you lots of interesting details. But what was more interesting for me is that the way in which they imagine and produce knowledge was pretty much like racialized and simplistic. And when it's kind of transformed into a law. The law had no teeth. So it was a beautiful idea, but actually the issue with this is that the Mexican genome is basically impossible to isolate. So there's no way to say this is our own genetic Mexicanness. And it was even more difficult because 25 million Mexicans live in the US, which makes it basically impossible to protect. However, the obsession has been to produce legal regulations that are supposed to protect this traditional or indigenous knowledge, or in this case, the Mexican nation. And that kept me thinking for a while. My main interest was on how race and nation shape science in genetics. And in this shaping of science, we produce a notion that we call it biocoloniality. So how actually these colonial legacies are shaping our political imaginations of the future, and how it shaped the knowledge we produce about it, about diversity, about race, and in this case, about how to protect that diversity. And then let me jump to the case of the poison dart frog. So it starts here in Mexico, the interest. So I have to explain you how and why we are doing this research. But really what interests us is the poison dart frog. So the poison dart frog is what we call the classic example of extraction of chemical value. So the important thing is not indigenous knowledge per se, but actually getting to know how indigenous people use, in this case, the poison dart frog to hunt in the blow, this little blow, I don't have the word now with me, blow guns. Yes. And you have these paper, like 70 pages of the guy that was doing this research, explaining in no detail how indigenous people use this. But then, and you would think he's an anthropologist of sorts, but really it's just to show the biochemists the potentialities of this animal. And once they collected 700 poison dart frogs and skinned them, they can isolate a little bit like milligrams of these poison. And the travel, I'm not going to go into all the beautiful details, but the travel of this chemical and how they found it in different animals until they discovered with the next breeding of frogs in the US that it was not produced by the frogs, but what they eat. They had this capacity to isolate the poison in the ants and in the insects of the Choco jungle to make this very potent poison. So the story we call it the story of purity, right? Is the story of valuing indigenous knowledge only because there may be some interesting biochemical that you can isolate. So the whole issue is not really to engage with the knowledge, but to use the knowledge as a proxy to find this important object that can be isolated in the biochemical laboratory. And we think this is classic biopiracy, right? But it's also classic science. It's not that they are doing something that is kind of like fundamentally wrong. That's the way they think they are contributing to society. And when you speak with many of these scientists, and then I'm going to go to Colombia, because this is not only a question about like, oh, the bad global north and the good global south. This happens within the countries as well. Okay, so this is not the classical issue about biocolonialism. It's about thinking how knowledge kind of shapes the possibilities for engagement. So what happened here is a classic case of purity. And we want to contrast that and think with another case, which is ayahuasca and hallucinogens. How many of you have heard about the trip treatment in New York? Yeah, that's very trendy now, right? If you go and find about New York University and Johns Hopkins, you're going to find that they're using LSD to treat terminally ill patients. And they have been having lots of success with psilocybin, which comes from actually magic mushrooms from Mexico, and has been known for millennia in the Mexican indigenous communities to be a very potent way to deal with illnesses. And still very widely used, by the way. This is not something that happened, and now it's gone. So this is the opposite of this classical biopiracy example of how the indigenous knowledge is erased to just get the chemical. And once you get it, then it doesn't matter. It can travel around, it can shape research agendas. Here, they found out that when they had some sort of shaman with them, and I'm going to call it a shaman, I'm going to give myself the flexibility to think about this as a shamanic practice, because actually a psychologist that follows the patients as they have a trip, their success rate was a lot better. So it's not about just the chemical, it's not about the psilocybin, I cannot pronounce that, sorry for that, my English is psilocybin. It's not just about that. It's about the indigenous practices that can travel with this treatment. And we thought that this is a completely different engagement, right? It's not the issue of erasing knowledge, but is the issue of bringing some of that knowledge to inform science. However, as you might know, that psychedelic research has been under fire for a lot of time, and these new clinical trials are really rare because for sometimes they were claimed to be very hippie experiments, and then they become classified drugs, and everything was stopped, even though it was very promising. And now the scientists have to be very clear that nothing of this is new age. So they have to make a huge boundary between what's going on, the clinical trials, and these other indigenous new agey others. However, already in the practice, you can see that they are importing, they're thinking, they're experimenting. And if you ask me, I find that much more interesting in thinking about how an indigenous bioscience could look like. And those are the questions that I would like to think with you. This is one of the quotes, the last quote, that is pretty much what many of the indigenous ayahuasca takers are looking for. So you can transcend yourself. You can see yourself as part of everything. And this is consistently what people participating in the clinical trials describe in this treatment happening in New York University. So why I'm telling you, I mean, we have, all of these I should have mentioned before, comes from a research project I'm doing with a colleague in Germany. So we have already drafted a research proposal because we think that these are questions that cannot be solved historically or ethnographically. We need to experiment with them. How would an indigenous bioscience look like? And how can a very thorough engagement with these two substances could inform the policy of the future, but also the practices of scientists and indigenous people in the future. So you remember that I started with the Mexican case, right? And how it was impossible to defend the Mexican genome. It is impossible, really. I mean, even when you have this DNA ancestry test, what they tell you is 42 markers, all right, out of trillions. So it's actually not so accurate and doesn't really defend anything that is of value if you're thinking about medicines and about the relationship between genetics and phenotypes. And it's similarly complex to try to defend and legally protect biochemical knowledge or indigenous traditions, because it's really hard to say what is the object you are trying to defend. And there's the other issue about all the money that is spent on litigations. And basically it means that you produce an endocolonial system in which just the super well-funded scientists inside Mexico can be the ones fighting for these sort of issues. And within the countries, there's also the issue of colonialism. And I'm going to bring two examples, one in which in Colombia is called the Great Human Expedition. Anyone have heard about the Genographic Project before? No, about this interest in the 90s about sequencing all the DNA of indigenous people to keep them for posterity, because basically they were going to be disappearing. And what we need to keep is their DNA. Yeah? No? Doesn't ring a bell? Well, of course, indigenous people were angry. I mean, we are not museum pieces, they were saying, and they were right. Why would you just keep the DNA of indigenous people? What was the point of that? Is indigeneity a DNA issue? Is your indigeneity there in the DNA? However, this controversy in Colombia and around the world, you can read, there are like 3 very good books. One of Jenny Riordan, that's called Race to the Finish, about genographic. There's another of Professor Madame Charek in the University of Amsterdam, that it also looks in real detail what's happening in the labs. But in Colombia, they had the same thing, because Colombia was celebrated as a mestizo country. You know what is the mestizo ideology? Have you come across that notion before? Ever? No? Mestizaje, admixture? Yeah? So basically, in Latin America, we had this notion that being admixed is nothing bad. because in the 19th century, being mixed was like, well, your race is actually degrading. And in Latin America, we created this narrative about admixture as being something good. So it's a pretty much racialized notion of what Latin America is, but it's in a good lens, right? So these Latin American countries, such as mine, Mexico and Colombia, I have to say I spent a year in Colombia doing ethnography, have produced this narrative. But indigenous people were having the same issues there. Like, why would you keep our DNA isolated? So they asked the scientists to stop these biobanking efforts. And the scientists were completely compliant. It was not even a controversy. So as soon as this happened, they stopped. And it was a very unfortunate kind of event, because the scientists were really eager to develop a fantastic program, and they were really holistic in their approach to indigenous communities, but they made the same mistake of thinking that the valuable thing to keep of indigenous people is just their DNA. So if you see it similar to the poison dart frog, right? What you want to keep is this chemical thing that you can isolate and then put in a museum for forever, or well, as long as humanity exists, or I don't know. And then what happened was that all the DNA database that they had collected, that also had medical records that many indigenous people had actually consented to and were working in kind of in tandem with scientists, was lost. And I think the lost was something we need to think about. We need to think about what is the product of these controversies. And if actually all the laws that happened subsequently after this controversy happened really change anything. My bet, after maybe 17 years since this happened, is that they didn't. That the law is just there sitting around, but it hasn't changed the relationship. And then let me go really quickly to the Waurani people we were working with in Ecuador. So this links back to the Mexican genomic case. So that's why it started. So they heard about this, and the Wauranis had an issue that some guys went to their communities, took a blood sample, and then they immortalized their cell lines, and their DNA is now part of lots of different, you can see there, how the samples were named, and they're now part of the collection of the Correal Institute. Of course, they never asked them about this. I mean, these are contemporary cases. This happened 15, 17, 20 years ago. It's not something that happened, as I tell you about the poinsettia in the 19th century. These are very recent cases of biopiracy. And the same issue happened. I work, as I told you, with the Wauranis, and they were trying to sue the scientists in the US for taking their blood and their DNA and patenting it. But of course, they don't have the resources to do that. And that means getting all worked up with the legal system of the US and trying to figure out how to go to court. So basically, even though they're trying really hard, probably it's not going to amount to anything. And it brings the issue back to the first question about sovereignty. And so they contacted us and started thinking, how could they produce something that is not about just legal punishment, a regulation, and all these notions about protecting the indigenous from the Western. Because as indigenous bioscientists, they were also genetic scientists themselves. So what they wanted to do, and I think this is where it becomes really interesting, is to produce biomedical science that could translate to different projects and laboratories around the world. They work with the Max Planck, with Sapienza, but also they want to make sure that these transactions do not mean that they are just contributing DNA and becoming the providers for other scientists. And this is, I think, where we need to start thinking about what comes after biopiracy. And I think historically, because of the way plants and animals have traveled, and also the human DNA has traveled, we haven't been able to develop any frameworks that can deal with this complexity. Because it seems really, really hard to show and to demonstrate to a court how indigenous science was key, for example, for isolating eripatidine, which is the chemical in the poison dart frog. Because it was only done thanks to the laboratories in the global north in the US, through all the experience of these biomedical scientists and chemists. So I think they have a claim to say, well, indigenous people knew about the poison, but we were the ones that isolated it. Therefore, what we have is different from their knowledge. And what that produces is, of course, loss of distrust. And this has actually made very difficult to advance these relationships that have been so important for the construction of biomedical knowledge today. If you think about psilocybin, it's because scientists working in Mexico and the US have always exchanged, but also indigenous people. There's a long tradition in Mexico about that, about ethnobotany that is really transformative, and it has really changed chemical compounds. And many of those things are found through engagement with indigenous scientists, indigenous people that know about these two worlds. And the other issue is, how can we come back to the problem of indigenous cosmologies and science? Is really the important question that indigenous people participate in science, or that minorities participate in science, or that the cosmologies can also be part of science, and science can be integrated into the cosmological practices of indigenous people. And I think that's the most difficult. I mean, for sure, more of us should be engaged in academic institutions. I mean, the graphs were telling about how difficult it is for minorities to be part of universities. I was reading that just now on Twitter that 4.2% of professors are female of all the population. So just 4.2 professors are female, which is it doesn't make sense. I mean, coming from an anthropology department where I work mostly for female, my bosses are female, and it has been my experience so far. But as soon as you get out of this little niche, then you find that that's not the case. Then these numbers are quite revealing. But maybe we need to go a step down the line and think about how indigenous knowledge could work with bioscience in a more horizontal way. And how to do that is the real question for us. And the problem basically is that we tend to legitimize knowledge through the lens of science. So all the knowledge that is legitimate has to comply with certain ideas, fixed ideas of science, which might not be the case. I mean, great scientists are not very compliant with the scientific method to begin with. But let's say that this is something that hinders a relationship between indigenous knowledge and science. And the exploration is also about ethics, about how natural history, so looking at these two examples, this is research we are hoping to do in the next coming years, how these two examples could inform new practices I have like 5 minutes left, new practices and forms of engagement between the Waurani scientists. So what we're going to do now is try to build a DNA database that has indigenous cosmologies, plants and animals, side by side with scientific standards and experimental ideas. So how we can hybridize this, and the thing is that we cannot do this historically or etnographically, we need to do it in practice. So probably the next steps for us to think across boundaries is to do participatory action research, which is building the things in themselves, and then thinking critically about this history. And this is basically what we wanted to do, and that was the point of the talk, to show you about the Mexican genomic sovereignty, how it has transformed the logics of biopiracy in Latin America, and try to maybe think and discuss, if we have time, what are the next steps, and what do you think about that idea. That's all. Thanks a lot.
Good afternoon. Can you hear me? And it works, right? Okay, good. I've got the great post-lunch slot, so hopefully I'm going to be able to keep you all awake. I'd like to thank you for allowing me to come and speak to you about what I do and about this program called Destination STEM. However, the talk I'd like to have is a much broader one, and it's about looking at the power of positive action. And I call it the power of positive action because I think it really is important for us to understand what type of enabling environment we operate in, and the fact that we do have agency to change things. So I always think it's helpful to give a bit of a background as to why I'm here. I grew up in London, went to state school, and I was the first person in my family to go to university. And I kind of always had this thing that we had to give back. So I kind of going to talk about quotes in a bit and mottos, but I've been in equality and diversity for 20 years. So I've been working in a space, either in the voluntary sector or in higher education for the last 20 years. And I've been in the Windsor Fellowship as a staff member, but I started as a student when I was doing my first degree back in 1996. And I was sponsored on a leadership development program. And again, that principle of giving back, making sure that the drawbridge is left open for those coming up behind me, became one that's very important to me. I then went into higher education. So I've worked at Cambridge, I've worked at Oxford, Birkbeck, University of London, and I'm currently the Gender Equality Manager, or the Athena Swan Manager at UCL. I'm just going to ask you, have any of you heard of Athena Swan? Great. Good. If you've heard of Athena Swan, I'm going to just say, I think it's a great thing. It's a great thing because it does take a lot of work. It does require a lot of people to give up their time and commitment. But it is perhaps, for me so far, the most effective structural change tool that we have in terms of equalities work. It doesn't just look at deficit modeling women. It does try and get the institution to try and identify the processes that it needs to change. And that, for me, is very important. What I'm talking about is a positive action, but actually structural change is actually much more fundamental. So I always say there's a balance between what I call the trophies and the tankers. And the tankers are much more difficult to change. In my other hat, I'm also vice chair of the Higher Education Race Action Group. So whilst I work on gender equality, I'm equally committed to race, and I run a race equality charity. And so I've spent 20 years giving information, advice, and guidance to organizations about what they can do to change things. So I work with the Bank of England, UCL. I work with a range of different public, private, and voluntary sector organizations to identify what are the positive actions they can take. So, and I'm also, because of this, been asked to sit on a number of committees and advisory groups. So I sit in the Royal Society of Chemistry's Diversity Committee. I'm of a certain age, so I was saying I like mottos. I always like things that are going to stand me in good stead, give me a bit of guidance. So I like coins. I'm a bit of a coin collector. So whenever I'm training, I always say, standing on the shoulders of giants, my £2 coin. And if you have the old 2p pieces, they say, ich dien, I serve. I always find that. But I'm of a certain generation that remembers Knight Rider. Yeah, do you remember Kate? Okay. One person can make a difference. Okay. One person can make a difference. And my question and challenge to you all is, what difference will you make? We all can make a difference. So for me, I've kind of gone equality-minded, so it's not one man can make a difference, but one person can make a difference. So moving on a little bit, he says, okay, I'm going to talk a little bit about the work of the Windsor Fellowship. Understanding positive action, because I think one of the challenges is that people are often frightened of coming near initiatives that look like positive discrimination. And I'll talk about that more fully. And then I'm going to talk about an example in practice, which is Destination STEM. So the Windsor Fellowship came out of the riots from 1981. So the fears that people had that we were going to end up having race riots in the 1960s, which then got people like Enoch Powell talking about rivers of blood, people were frightened actually in 1981 post those riots. Oh my goodness, what have we got ourselves into? And so there was a conference as part of the aftermath of this at Windsor Castle, which is why we take the name Windsor Fellowship. And in that conference, there were members of government, business, and communities that were saying that basically people from black backgrounds, and I say black in a political sense, do not get access to opportunities. And as a result, a charity was set up to be a bridge. And bridges aren't comfortable things if you think about them. They try and link two sides together, so you can often get a bit worn out. Okay, but they're important. Without bridges, you can't create communities. It's much more difficult. And Windsor Fellowship was set up to do three things. Improve educational attainment, to make sure that people could make the most out of the educational opportunities that we provide. Develop active citizens and active citizenship, There are a lot of people who are out for themselves, but if we don't understand what community is, we have a real challenge as a society. And active citizenship is about actually, how do I contribute? How do I make my difference? And the final part is about translating those two different preceding points into something which is called career-focused employment. There are lots of people who are very bright, but they've been frustrated. Lots of very intelligent women, if you go back 100 years, who could probably get the best degrees and qualify the best, but they weren't able to get the degrees. So I worked at a university at Cambridge. It's a great university, but they only started awarding women degrees in the 1940s. It's over 800 years old. And so in our society, we just need to recognize we have lots of institutional and structural barriers that prevent people realizing their potential. So what we need to do is think about how we can challenge that. So that's what the charity was set up to do. And we did that by focusing on a concept called positive action. When you have these structural barriers in place, there is no point in saying we'll just stick an advert in a newspaper and say, look, everybody can apply. The social capital, the knowledge just isn't there. So we need to think about, actually, what can we do proactively to change things? Now, change doesn't happen overnight. Our legal regime, and for this, I'm quoting the Equality Act. So the Equality Act brought together lots of legislation. I think over 200 pieces of primary and secondary legislation were drawn together. And we have positive action provisions. Now, positive action is not affirmative action or positive discrimination, but it does allow, but does not require, any action that is a proportionate means of achieving a legitimate goal if it helps to support those having a protector characteristic overcome barriers. And that's to do with education and training, but not employment. So you can run training programs, internships, work placements, okay? And there's studentships might be part of that. And organizations that do Athena Swan, so I saw a very large number of people who are familiar with that, will know some of the positive action initiatives. And this could include mentoring, providing specific training, and there are a lot more comfortable initiatives that are being done around gender because we're much more comfortable dealing with gender, if that makes sense. Nothing wrong with it, by the way. This is not a competition. Really, it's not a competition. But it's about sharing that learning and developing that competency in one protected group for another. And that, in this particular case, we focus on race, but it could equally be disability or sexual orientation. The law is the same law. That's the benefit of the Equality Act. Now, what does that look like? Well, essentially, it's about preparing people. So, while I said we have the same rules now and everything doesn't look different, it all looks the same. It's about making sure that people can get to the starting line of a race, if you think of it this way. So I see it as a race. If you see it as a competition, because they have a recruitment competition, getting people the training and the support and the guidance to know what the rules of the game look like so that they can make competitive applications. And what we're trying to do in that is enabling people to understand the breadth of opportunities that there are there for people. Now, we need to have a bit of reasoning as to why organizations might want to do this. And sometimes you don't always have the data. So if you look at certain communities where we don't have a lot of data, okay, we may not be able to say, well, definitely, but you can get a sense within an organization, particularly if we look at gender, we look at race. Disabilities, there's also some data there that shows that there is an underreporting. So we have an underrepresentation. So it's just, you have to have a reasonable expectation. You must reasonably believe that there's something that we need to change. Why wouldn't we do that? And that's how I always find that one of the challenges. Why would organizations which can see underrepresentation not make an effort to change things to be fairer? Now, when we talked about Destination STEM, I worked with colleagues to identify and look at some of the data. Now, one of the things I'd say, I'm really grateful that I work in Britain. We're good at data. So for those of us who work in higher education and are familiar with higher education, we have the Higher Education Statistical Agency. They produce lots of data. And if you want to find out about equalities data, there's a subset of data called HIDI, and it's not the story from Switzerland with the person who lives on the mountains. It's a set of data, it's a data set. And if you work in higher education, there will be colleagues who can access that data. And they can tell you what's what. In addition, there's an organization called Advanced HE, formerly known as the Equality Challenge Unit. They run the Race Equality Charter and the Athena Swan Charter. And they produce a statistical report every year, which you can download from a website. And it talks about some of the key equality challenges in the sector. But what the data behind was looking at was where people were studying in STEM, or science, engineering, and technology, okay? And you can see that certain groups are likely to study certain subjects more or less so than others. So it's just trying to use the data that we had. This data, by the way, is a couple of years out. There is more recent data that will be available, okay? But the data is indicative. It's about understanding the point and the principle. So we took the data. And what we wanted to do from this is to say, if we want to change the way that the professorship looks in universities and our leading researchers look, we needed to think about, actually, what's the pipeline? What's the run-in? I've been speaking to colleagues here who work in museums. To work in a museum, you can't just stick a sign in the newspaper saying, please apply. It's a much more thought out, long, drawn out process that could take the best part of 15 to 20 years if you really want to do it in a serious academic way. And so what we need to think about is, where do we start? So I was approached by a colleague working at the Royal Society, and they were looking at this idea. And they were looking at this data because there'd been a report that had been produced, and they were then saying, well, what can we do? And so we decided that actually we wanted a long-term intervention and I say a long-term intervention by helping to start something along very early in the pipeline. So I know colleagues, when I say Windsor Fellowship, we provide programs, so we do something called the Junior Fellowship. So I know a colleague in the room has done the Junior Fellowship, which works with students in what is now 4th or 5th year, which is now years 10 and 11, okay? And we work with programs doing A-levels, so years 11 and 12 and 13, okay? And so what we decided to do is to provide a mentoring program for students in years either 12 or 13, and I'll explain why we've changed it. We started with year 13 to begin with, okay? And we reviewed it in the second year, and we moved it to year 12 so that we could influence things. But the purpose behind this was to help A-level choices and to enable participants to have as much information about STEM as possible. And what we did is we thought, well, a one-year program, we'll start in London, we'll pilot it, we'll look at A-level, people interested in A-levels or equivalents in STEM subjects, and then we'll tie them up with mentors from the Royal Society. So the Royal Society helped to identify people who were willing to be mentors. And those people decided to give up their time to meet up with our students every month. They provided information, advice, guidance. Some of them even took them to brain-cutting departments within their institutions. They brought them up to see mentors, meet their PhD students, took them to lectures. And the idea was, how do we develop the STEM capital in these young people so that when they come to think about careers, they don't just think about the vocations. Because some evidence is out there that many people from BME communities, when we're looking at jobs or careers, we will opt for vocational subjects. And that's partly because, one, we're aware of those jobs and careers, and two, because if you're worried about unemployment, you might think, actually, this is a sensible thing to do. Give yourself something that's going to give you a job. It's safer. And so what we decided to do is, how do we help people understand the full breadth of STEM? And that's what these wonderful mentors do. And they are really wonderful people. I meet them and I train with them and we work out what's going on and what we can do to make things better. The other part is to look at parental engagement. So we work with parents. And one, there's a very practical safety thing. You want to reassure the parents that, one, we've done the due diligence, we're taking them on this program. But more importantly, it's helping them to understand that, well done, your child is doing well, they're on something positive. Because there's so many negative stories out there about young people at the moment, so it's good to tell parents, you know what, you're doing a good job. come and we take them to the Royal Society on the introduction evening. There's a real opportunity there. And this is because we had the idea that you have to work with parents in order to help them figure out how they can then change and support their young people as their role changes, because they may not be the primary source of learning and guidance. And then to complement that, we also run training seminars, skills development seminars. And the idea is about thinking about how do we help our young people understand what soft skills look like, whether it's presentations, interviewing, thinking about project management goal setting. Because part of this is about thinking you've got these different parts of the journey, which are, we want advice and guidance and information. We've thought about courses and universities, but actually how do I set myself realistic goals? And how do I then set myself project plans to get myself on those goals? And we use a lot of volunteers to help us do that, and we pay people to do that. And that is what this Destination STEM program is. It's a really good program. In our first year, we set up 23 mentoring relationships. So this is the first year. Over the 9 months that we measured, we had 326 hours of mentoring. 21 out of the 23 students went on to STEM subjects. Okay, 17 of them at Russell Group universities. Yeah. I'm often being told that black kids can't or don't aspire. That's not true. I don't buy that. And I often find people tell me all sorts of weird and wonderful things, but actually, if you provide opportunities, people will thrive. And these people are. And there's a really good partnership there. So much so that even the mentors, going on to the second year, 14 of the 23 mentors came back to mentor in the second year. When we've been piloting it, we're on our third pilot this year, okay? And what we've learned is that we wanted to move the program a year earlier, so that we started with year 12 students, because that actually helped us to inform A-level choices. Because colleges are quite challenging places to change courses midway through, so we thought we'd get in as early as possible. We even have an even narrower window at the beginning of the year when you have flexibility for course changes. So what we did is we then thought about, actually, we need to get this earlier. So we've been doing this now we're on our third cycle. Thinking about the impact, okay. When we looked at STEM Capital, we're trying to figure out actually why are we doing this program? Well, a bit of evidence. It's anecdotal, but it's evidence that we've figured out. At the beginning, the knowledge of STEM, scientists, okay, has increased. So between seminars 1 and 4, we've seen an awareness of STEM and what it can do. And this is just about naming people, okay, so 3 males, 3 females. Some of the shadowing I find is fascinating. The bottom picture there, the brain lab I was talking about, the bottom picture on the right. I have to be honest, I found that a bit queasy, but it was the students loved it, and it became a source of a lot of pride that they were going off to these really interesting places. The photograph on the left is from our welcome event, and the Royal Society made an investment in the program, and they bring the students and the parents together to meet each other, but also to meet the mentors. And it's about recognition. You're doing something which is respected and esteemed. And one of the other things is about creating a network of peers. Because if I have a problem, actually, I may be the only person to experience that problem, but if I have a network of friends who are similarly interested in science and who might be interested in taking this work forward, we can work together. And actually, they WhatsApp each other, they support each other, and it's become a space for them to meet up. And 2 years on, we regularly still meet up. So, again, it's just thinking about what the impact was. And I think there are lots of other quotes I can give you. One student this year wrote about how it felt wonderful to have somebody who actually believed in me. I may be in school and I don't always feel that my teacher believes in me, but actually I've got somebody who comes and who's got the time for me and makes the time for me, who knows what they're talking about. Some of the mentors have actually been relaying to me their experience of what it's like to work with young people, because they're scientists and they may be going on to be PIs, and they're saying, principal investigators, and they're saying, well, actually it's really nice to be able to work with somebody that you might have to kind of manage in a way, and it's kind of giving me a forerunner into how I'm going to develop my skills to work with other people. So there's a really useful relationship that they're getting out of this. And it shows because they come back and they've contributed, then they're invested. So, I remain grateful for that. These are some of the courses where the students have been going on. It's a small sample, but you have to start somewhere. Somebody asked me once, why, what's the numbers? Why are you doing this? I'm not sure if any of you have seen the data. There are, I believe, 70 black professors in the entire United Kingdom, according to the data. There was a report that was produced a couple of months ago that identified 26 black professors in the United Kingdom. We need to change. We have to change because, one, we're losing out as a country in talent, but actually social justice demands that we need to be doing more, and we need to be doing more faster. Because left to its own devices, things will not change. And that's why positive action was set up. We were given an enabling environment to allow us to see where a problem is and how we can change. So when I think about this, I'd say, don't be shy. It's easier to cower down and be frightened because there'll be lots of obstacles that organizations may raise. And I'd say, look at your data, see what the law says, speak to people. There's lots of people out there. We're one organization. There's lots of networks within universities where positive action, widening participation has already been taken up. Think about how we work with parents and communities. It's really important because, in a way, I was having this conversation over lunch about Charlie and the Chocolate Factory. In a way, universities are the modern factories. And what we don't want is that Charlie kind of mindset. You bring somebody in for a day and then you send them home. Because actually that doesn't necessarily help in terms of delivering social justice and change. What you need to think about is actually, how do I invest in the medium term? And recognize that there are benefits for all of us when we participate. And long-term change really does need sustained commitment. We are the inheritors of a really decent society overall, but there are lots of structural issues that we, one, need to acknowledge exist, and actually we need to invest to change. And that does take time, and that does take focus, and that does take commitment and investment. And it's not just the minority community, so the women can't be doing all the gender equality work. If any of you hear me speaking on this at UCL, it can't just be the women that do gender equality work. Okay, and in the same way, it can't be this the people of color, the black people, the Asian people sitting there doing the race equality work, because everyone has a responsibility and a benefit when we work and live together. That's the photograph from, again, I'll start that, and that was again, it's a great mix of people, and makes me very proud. When an organization decides to work together and work with a community organization, you can make a difference. And that's us. So if you want to follow us, find out more about us, drop us a line. I'm happy to answer any questions.
Well, thanks everyone for coming, and thanks Leanne for inviting me along. Oh, it'll be up here in a moment. There we go. Right, it is really humbling to be here, and it's humbling to speak alongside the other speakers who have spoken today. I'm going to tell you, it's a very specific story. It'll be a very specific story about Bristol. It'll be a very specific story that arises from Bristol's year as the European Green Capital. So the story starts roughly around the autumn of 2013, when we were announced that we would be the 2015 Green Capital, and it carries through to today. And it's very much about inclusion. It's very much about social justice, and of course it's very much about the environmental movement. But with that, I mean, not just sort of like the politics and the campaigners, I mean all of us. I mean the green industry, universities, museums, societies, the whole group of us. And so even though I'm telling you a Bristol story, and it's a very specific Bristol story, I do think that the lessons are very broad, very applicable, and you'll hear me hit on many of the same themes that we've been talking about and hearing about all day long. I suppose I would start with sort of say, it's fantastic that Bristol was the green capital. And of course, Bristol is known for being a very, very progressive city. And I would suspect that, really much of what I'm going to tell you emerges in this is often a bit of complacency that comes when you actually think that you have your social justice warrior badge ticked and your environmental justice warrior ticked, and you sort of think, we're doing all right. And you forget that even though you live in a thriving progressive city, it is a city plagued by economic inequality. We have one ward in Bristol that has the highest rate of university attainment in the country. We have another ward that is near the bottom. We have adjacent wards where you have a 10-year difference in life expectancy. And of course, this inequality did not arise out of nowhere. It arises in part because of structural inequality in the system, and of course, Bristol's wealth being largely based on the slave trade. And of course, many events in the green capital would have been held in buildings named after slave traders. Leanne got her PhD in a building named after a tobacco trader, Will's Memorial Building, where I have my job. And okay, Will's did not trade in slaves, but in Bristol, the tobacco trade of the 20th century is not that far removed from the slave trade of the 19th century. This is deeply permeated, and of course, the BME populations, especially the African Caribbean population in Bristol, is acutely aware of it, and when you're a well-meaning environmentalist, you don't get to ignore it. We did, when we became the green capital, we realized that inclusion was going to be right at the very, very heart of the mission. And by we, I mean a huge number of green organizations. Bristol is home to the Green Capital Partnership, which has 800 organizations. So I'm talking about a huge number of us. And we realized that inclusion was absolutely essential to what we wanted to achieve. And of course, this is now coming back to us today with the Green New Deal centering inclusion and a wide range of equality components in it. So this is, it's not new. It wasn't new then, it's been pervasive. And of course, there's good reasons for it. It's quite pragmatic. Why do you want to have an inclusive environmental movement or inclusive environmental organizations? Well, obviously, something like climate change is going to impact marginalized groups the most. We have seen this time and time again, so it's only right that we center their voices in the discussions about that. Obviously, if you want to have a successful movement of any kind, and you want to make a big change, such as eventually us moving off of fossil fuels or stopping the sixth extinction which we are experiencing. If we're going to make these types of fundamental changes, we need as many people as possible. I would argue that those are very vital and they're important, but they are somewhat transactional. And we sometimes forget, of course, the last one, which is that if the environmental movement is thriving, and of course environmental economy is growing, the environmental economy is growing a huge amount, so actually that imposes a little bit more responsibility, because if we are not opening the doors of that environmental economy, we are simply perpetuating inequality. We are now participating in it. And it is no different than if universities are not opening their doors to marginalized groups. It's a very, very similar thing. There's an obligation that goes beyond just the transactional of the previous points. But we recognize this, this was important. I would argue it was not structurally embedded in the organization that delivered the green capital in terms of who we hired and how we hired. We hired events managers. We did not hire the people who understood it. So we hired a lot of well-meaning people but did not truly understand what needed to be done. But nonetheless, lots of programs, lots of talks, lots of summits, all sorts of things, lots of art exhibits and things that were all over Bristol. There was an art scheme in every neighborhood. There was a neighborhood partnership program. There was no shortage of activity that did reach all across Bristol. And yet, nonetheless, as the year went on, and certainly near the end of the year, the pervasive mood of everyone, it hadn't nailed it. It just had not managed to overcome the barriers to inclusion in this city. And, that was because those of us who were involved with it simply had not done what we should have, and we'd missed the trick. And so part of this, there's basically that there were ongoing components. The Green and Black Project actually represents 3 components. There was the Green and Black Project, which was an ongoing discussion during the year to give voice and present environmental and sustainability initiatives led out of the green movement. That then led to the Green and Black Conversation towards the end of the year, which was probably a more critical discussion about where we really thought we should have done better. And then after that, we then had the Green and Black Ambassadors. So I'm going to tell you a little bit about the project, a little bit about the conversation and the conclusions, and then what the ambassadors represented. And the key components in all of these were sort of, so you had my organization. I was director of the Cabot Institute at the University of Bristol at the time, but really it was led as a partnership between the Green Capital Partnership, and in particular, it was Ujima Radio, which is a BME-led and run community radio organization in Bristol that is very active in all sorts of things, and predominantly comprises and reaches out and connects to the African Caribbean population in Bristol. And I'll come on to Zakiya and Jazz in a moment because they were the ambassadors, but I'll build up to them. So the initial bit was the project, and the project was solely Ujima's at the time. This was their passion. They worked with another community radio station, but the goal really of the project was simply, let's bring out these voices. Let's make sure all of the voices are heard. They led that initiative, and I think that was a fantastic thing. As I said, as the year went along, we realized that it was just not quite as inclusive of a project as the Green Capital, the whole thing was not as inclusive as we had intended it to be. And that's when we became involved with the partnership, and we provided funding to Ujima Radio to convene a couple of, as I said, more critical discussions to explore perceptions, the realities around what could have been done better. It was done in stages, and mostly today I'm going to talk about what we learned from the first conversation, and that was a conversation, again, led by Ujima Radio and some of their colleagues, to really open up a discussion from a BME community perspective, how they perceived how the year had gone. This second conversation, which I'm not going to talk much about, I found particularly interesting. That's when we brought that community together with sort of the usual suspects in Bristol's environmental community. And I do think that was actually quite valuable. I'm more than happy to answer questions about it, but I think it sort of revealed where some of the tensions come along. And all I'll say about that right now is I see the risk of some of the same tensions right now, and that is if you've been fighting something like climate change your whole life for 20 or 30 years, or if you've been looking at this huge mass extinction that we are doing to the planet right now, and we were looking at this huge biodiversity loss, and you've been fighting that for 30 years, you will get impatient. You will be intemperate, and sometimes you will just want to forge on and demand that action happen, and that is not helpful for inclusion, but it is understandable. But we can talk a little bit more about that, and we can talk about maybe how we run the risk of that with some of the language that we're talking about climate change today in terms of it being the most urgent problem, the most important problem, at the implicit exclusion of other challenges that we face. But through this conversation that was chaired by my colleague, and that was Roger Griffith, who is the founder of Ujima, or co-founder of Ujima Radio, these were some of the main conclusions. So there's going to be a lot of text on the rest of these slides, because obviously if the point of this initiative was to give especially the African Caribbean population voice, now you've got like white male privileged academic talking at you. What I've done is I've used their words and I've put a lot of those in here. I did try to record and video them a bit to sort of actually get them in the room. I am not savvy with the multimedia. But you will see if you follow some of the links, there's loads of sound clouds, there's loads of videos, there's loads of things where you can hear it right from them, and they are far more inspiring than I will ever be, especially Jazz and Zakiya, who I'll introduce in a moment. But this is from Roger's report. This was Roger's synthesis of what came out. So the first thing is, wow, we overused the phrase, how do we get BME populations more engaged? These communities
so many leaders, so many citizens, so many residents actively engaged in it. Just because they're not coming to my event or my university or my institution does not mean they're not engaged in that. So there was a real sort of sense of condescension that needed to be, that was embedded in our language that needed to be snuffed out. BME leaders were invited to participate, but often these were coming from voluntary organizations, and there was no financial compensation. They had very little resource. They were taking time out of their day. As an academic studying climate change, it was part of my job to go to these events, not someone who's running a small community radio station, or someone working for the Malcolm X Center, or someone working for a mosque, right? So there was no compensation, which obviously affects some groups much more than others. There was also an organizational component. Bristol sort of prides itself on its anarchic characteristics, but you know, if you're organizing an event at the last minute, do not expect, well, actually, you do not expect poor people to show up. Do not expect working class people to show up. Do not expect people with kids to show up if you're not well organized and you have your events well far in advance with proper, you know, creches, support, those types of services. If I were to do this whole thing over again, I would say Bristol should not have done anywhere near as many of events they did, done far fewer, but to a much higher degree, that are much more embedded and supportive of participation by people from all walks of life. Again, different abilities, different family contexts, different working contexts. The venues were often not accommodating. This one stung. One of the main places we had our green mingles was this fantastic restaurant called Bordeaux Quay. Why did we do it there? Because it was free. Why was it free? Because it was owned by the sort of, well, it was owned by like a little collective of hippie environmentalists, brilliant people. So it was free. But you know what? It was a rather posh restaurant on the waterfront. And again, coming back to sort of issues of intersectionality, but if you're sort of like, if you're not only from a marginalized group, but you're working class and poor, this was not a place that you felt you could just walk in and be welcome. There was these chronic issues that we talked about earlier, which is that those communities were already beginning to suffer a decade or so of a lack of funding, so there was a leadership deficit. And I think overall, there was the perception that there was a lot of talking and listening, but nothing actual real was changing. And I would say, so these were what Roger collected, I would say, this is a bit, none of this is new, right? We could have found this out if actually this had been embedded in how we had actually structured and organized the program in the first place. And I think this is my call out. So the Cabot Institute is an interdisciplinary research institute. So this is my call out. Most of us are probably scientists or scientist-ish. A little bit more time talking to some social scientists, some anthropologists, some people from the humanities side of things, would have revealed the deep literature and understanding about exactly these types of issues. So it's, you know, if we're talking about reaching out, we should also be reaching out to different forms of knowledge in different disciplines. We have a lot to learn from our colleagues. This is why it's great to have a talk earlier that really brought history and the humanities and the sciences together in such a beautiful, beautiful way. So based upon that, we said, well, what are we actually going to change? We just, you know, sort of that last point, we just spent, you know, we spend a bit of money to put on a couple of events, paid for some food, and we spent a lot of time talking about it. What were we actually going to do? And this is where we were able to get money from the university and money from the ERC through a grant and money from the NERC to fund the Green and Black Ambassadors. It was funded on a shoestring. We weren't able to, you know, keep it going for long. It was a pilot program. But the goal was mentor and empower new people, train them in media, science, citizen research. The longer term goal was a political project. It was to put them and Roger and Ujima Radio and other people that they were advocating for into positions of influence and power. It was basically to hold big Cabot Institute University of Bristol events in the Great Hall of Will's building, and then concede the stage to them. We have Festival Ideas, all sorts of big name speakers come on that. Bernie Sanders came, Festival Ideas said, we want you to chair the event. So there was a lot about visibility, profile, and giving them voice. And that went alongside their media training. And ultimately, it was to sort of challenge and disrupt institutions and make us question our behavior. There were some principles and values went into it. They were paid, as were the partner organizations. We viewed this as we don't have it sorted out yet. So rather than asking for them to volunteer their time to help us solve our problems, if you're going to go that route instead of educating yourself, pay them the money. So it wasn't just the ambassadors that had to be paid. We paid Ujima Radio for their time and their effort. The ambassadors, along with our partners, set the agenda. We supported, we facilitated, we put all the time in, but they set the agenda, and we tried to connect with venues all over the city. Unfortunately, what you'll see is that we weren't able to achieve anywhere near as much as we wanted because we didn't have all that money. We managed to scrape money together, and it is ongoing, but we've not yet been able to scale it up and grow it, unfortunately. Well, what did they do? Well, they did a lot of radio shows because Ujima is a radio station. They used the radio shows to profile other BME leaders in the environmental sector. There were lots of events. There was lots of discussions and interactions. I'm going to talk about a couple of those. And again, lots of high-profile events that other organizations ran that they participated in. And we also got them involved in various board meetings of some of the major green institutions around the city. So just as a couple of examples of the things they did a lot of evidence gathering. They went and talked to a lot of, again, African Caribbean residents. This is a quote from the radio show. And this is just like someone who phoned into the radio show. And again, it sends the same message. These communities, are invested in the environment. They care about the environment. They really do. And just, and you can see in the quote, they're pretty much imploring us as museums, as societies, and universities to reach out to these small community initiatives and engage them. They also used a methodology of co-production called photo voice, where they gave cameras to citizens and let them go out and take their own photos and try to capture their own story of nature in Bristol. And again, that helped open up further conversations. This is a really important slide to me because we were giving them a stage. All of a sudden, they became quite visible. And we certainly did not want them to eclipse all the other incredible initiatives that were being led out of different components of the BME communities in Bristol. Part of their job was to interview people like this for their radio show, profile them, lift them up. And there's all sorts of people. There's Cleo Lake, who's a, I always mess up with her Twitter handle. So there's Cleo Lake. So she is a green councillor in Bristol. She's now Bristol's Lord Mayor. Over on the far left, I don't know if any of you recognize it, but that's Maya Rose Craig. That's Bird Girl. Have you heard of Bird Girl? She's the youngest person to have ever seen 5,000 species of birds. But she has a very prominent Twitter feed. She gets a lot of attention through this. She uses it to address issues of lack of diversity in the environmental sector. She led a great project called Black to Nature, which was, again, that was about getting inner city students out into nature. Simply by using her platform and her reputation in the conservation and the wildlife community to simply say and talk about issues of diversity has often attracted truly, truly horrific attacks on a 14-year-old. And her bravery sort of reveals, some of the challenges that we face that are embedded in our communities that let's face it, that we like to think should be better, right? So I think that, so there's all sorts of amazing people that they profiled. And then the last, oops, sorry, I went the wrong way. There we are. And then this last one, they did do one bit where they really dug in. They worked a lot with the Slimbridge Wetlands Center. So they had a couple of workshops. They went there. They sort of talked about it. They talked about why it's difficult for, again, in particular, this was aspects of the African Caribbean population in St. Werburgh's and St. Paul's. And they raised issues like cost, not having the right boots and coats to go out into nature, various things. What I loved about this story is what it did is, not only did it raise these issues, but it opened up this conversation. So they did one visit, then they had this workshop where they sort of spoke through it, and then they went back again. And I want to say, will anything change? And what Slimbridge did was just a subtle thing. You can read it for yourself. What they did is they created a tailored experience, and what they did is they created this bird bingo game. And what they did is they realized is that many of the people coming had come from all around the world. The students, the young kids, they might, they might have diverse heritages, but sometimes their parents were immigrants who had come from different parts of the world. And what they did is with this bird bingo, they connected the birds that you could go see just half hour out of Bristol to birds that would have been from their homelands and the countries they had come from. And I thought, just that little gesture had huge resonance and power. And again, so there's big schemes you can do, but these gestures of solidarity and connection, these human gestures actually made a huge difference. And they found that very empowering. So I'm just going to wrap up with just showing a couple of pictures of the actual ambassadors and then their own words. This is Zakia. So we were only able to fund 2 ambassadors. We're hoping to get lottery funding to fund a couple more. But you see, these are very, very powerful statements from Zakia, right? This is a confident young woman. She's talking about, the challenge and disrupt, but also support existing institutions that change our behavior, to address unconscious bias, discard practices that are implicitly exclusive, and to proactively think about how to create structures that are more inclusive. This was a quote. She recently was awarded a Forestry Commission Writer-in-Residence. This is a quote she said along with us. "We are often the uncomfortable ones. Wildlife organizations need to come to our territory and be uncomfortable too. Meet us where we are. Come to our meetings in our space and ask us, what can we do? What are we missing? I need to get people from my communities out as well, which we're trying to do." It's about meeting in the center. Jazz, she also has these great quotes, but I decided to include this sort of what I thought was a story that she told, and she kept coming back to that I thought was quite powerful. They wanted to connect up with the elders in some of the communities. So she wanted to connect up in particular to elders in the Somali community. I'm just going to start reading from about here. So she went and talked to one of the more esteemed elders in the community. He said that the elders are asked to take part in research a lot, or at least people would come and talk with them all the time. But they never seemed to give anything back. He explained it felt like they were being used, and there was never anything returned to them to say thank you. The community leader wanted me to meet with the elders regularly, organize meals and activities, and then over time carry out my research project on elders. Given the time constraints, she couldn't do that. And this simple story has all sorts of lessons. First of all, I've been kind of flippantly throwing around BME communities. I kind of flippantly throwing around the phrase even African Caribbean community. Jazz came from, came from a community. She was not a member of the Somali community. Not all BME people are the same. And there's a multitude of communities, and there's a constant effort to build dialogue and connection. And that's the second part of her story, of course. It's incredibly powerful, right? It's asking for time commitment. It's asking for effort. It's saying put in the effort to build trust, which obviously resonates entirely with what we were talking about earlier. It can't just be bring someone in for a day and send them out. It takes effort. And the elder is not saying trust specifically there, but he is clearly talking about trust. Build trust by investing time in us. And I found that to be just a very, very powerful story. Just as a final closing thought, and again, I'm happy to answer any sort of questions about the mechanics of it, which I've rather glossed over, and also ask questions about how we try to keep it funding and get going. But I just sort of love this final quote. This is again from Zakia. But basically, it's like, it is a big task, but as she says in Jamaica, one cocoa fill basket. So, and she, and that's how she sort of approaches a lot of things in life. And so, I suppose, to again come back to points that have been made a couple of times, it's right to do this. It's the right thing to do. It can be very, very hard to do at times, but it's rewarding for everyone involved. And although I was technically meant to train and mentor them about climate change science, I learned far more from them than I'm sure they learned from me. So thank you very much.